show you how we can extract genes IDs from BIOMART. BIOMART is one of the most, I would say, revolutionary development in the field of genomics. BIOMART is embedded into uh, ensemble, and you can extract bulk data. One of the most useful tool to extract the bulk data from internet. So first of all, this is Google. Just write BioMart here, um, and this is Ensemble BioMart because BioMart is embedded into Ensemble Genome browser. You know, so let's we use this international website of Ensemble because it's faster. So here is BioMart. So in BioMart, first of all, you have to choose which database you will use. So we will use Ensemble Gene 108. This is latest one. And then which species the genome of which species you want to select. So let's say we want to select human genome. Just this is just an example. It's up to you, whatever genome you want to select. I am at the moment going for human genes, but you can, I mean, if you have mouse genome to focus upon, you can go for mouse or rat or zebrafish, whatsoever. So this is, I selected human genes, and then this left column, this is a very important column. Here, first of all, there are two terms, two tools, I would say, which you should know. One is the filter, and the other is attributes. In the filters, you add your query. What is your query? And in this way, for instance, because we want genes IDs of mice genome, let's say we go to gene and we go down to, let's say, gene type. So let's say we want all the coding genes IDs of a mouse genome which are present in ensemble, and let's say yeah, this is protein coding genes. Fine, the protein coding genes because we want to get the gene IDs of protein coding genes. And then the second part is the attributes. In the attributes, you want to actually select what information you will get in the output. So in the output, there is features. Features in the gene, for instance, we want the ensemble gene ID. We don't need the version ID. We don't need the transcript IDs. So this is ensemble gene ID. And the second thing you also want to know the gene name. So gene IDs and gene names of the coding genes of mouse you want to get. So in the attributes we have gene stable IDs and gene names, while in the filters you have gene type protein coding. Now go to the results, and you will now save it. The best option is to save it in the form of Excel because this is the Excel data base. I mean Excel sheets which you will use for your later, usually for your later data analysis. We go to go. So let's be in desktop. Here it has been saved. Results have been saved in the desktop. Let's we open it. It's opening now. taking a little bit of time because it's a heavy file. So you can see here, sorry. So you can see here, uh, we have this data. Now the result. These are gene stable IDs, and then there are gene names. These are the gene stable IDs of mouse genome, the protein coding genes, and then there are gene names also. In this way, you can see here, you know, now have all the protein coding genes of mouse, which is around uh, 22,896 protein coding genes IDs you have collected. So in this way, you can very easily, with simple clicks, by using your laptop, you can simply get all the gene IDs of the protein coding genes of mouse or any species you like. Thank you very much.